Sí, buenos días a todos. Gracias por estar acá. Bienvenidos al 18º Congreso Nacional de Ciencia y Tecnología, APANAC 2021. Mi nombre es Javier Sánchez-Galán y esta es la sala magistral del primer día, el día miércoles 23 de junio. Antes de comenzar, pues quisiera agradecer a los que estuvieron ayer presentes en la inauguración del Congreso, dar un especial y grata bienvenida pues a los que también nos vieron a través de la plataforma, ya sea de Webinar Jam o de InContacto, del Feriale. Y solamente tengo unos par de anuncios, algunas cosas que sé que están en la mente de muchos. Por ejemplo, los accesos, algunos de estos accesos se están ya completando. Sobre todo también quería haber recibido preguntas sobre cómo es la agenda. Pues pueden ir a la página web del Congreso, congreso.apanac.org.pa, e ir a la sección. Hay un botón grande que dice el libro del Congreso. Se ha preparado un libro del Congreso que tiene toda la información relevante del Congreso, los números, los nombres, las ponencias, y pues se encapsula todo lo que es el Congreso. Pues, y en el día de hoy en la sala magistral tenemos dos charlas fenomenales, pues también, digamos, invitados pues de la Embajada de Israel, y voy a presentar la primera de ellas. La primera charlista va a ser la doctora Yonath, del Center for Biomolecular Structure and Assembly del Weizmann Institute of Science. Ella es Premio Nobel de Química en 2009 por estudios de la estructura y función del ribosoma, obviamente entendido pues que esta es la maquinaria celular de la creación de proteínas, un problema que era bastante complicado y que ella, pues, a través del tiempo, pues, pudo ir abordando, lo que le llevó a ganar el premio Nobel. Bueno, con ustedes, sin más preámbulo, pues dejo a la doctora Yonath.
I would have liked to be here today with you, but since I cannot, at least I sent my lecture, and I would like to tell you about what is it. It's about the translation of the genetic code into its products, the proteins, and how one can stop it if one wants to stop the life of a pathogen by antibiotics. Antibiotics are fantastic, but there are problems with them. The main problem is resistance, and we are trying now to fight against it, and that's about what the lecture talks. Thank you. I want to talk about next generation antibiotics, or at least what we suggest should be next generation antibiotics. And for this, I also want to show my motivation, which was actually curiosity, like that of our hero, Mr. Einstein, that said that all what he had was curiosity and passion to it. When I became a young scientist, I thought the translation of the instructions embedded in the genetic code into proteins is one of the most interesting life process. Therefore, I focused on it. What do I mean? Let's first see what proteins do. And what I use here is some pictures from a children's book about proteins in human. But actually, proteins perform nearly all the essential functions in every living cell, even in bacteria, in flowers, in trees, and in human. So here there are some proteins. Hair and skin, structural proteins, signaling proteins like insulin, transport proteins like hemoglobin, sensory proteins like vision, and enzymes that are taking apart the proteins that we eat to their components in order to make new proteins, those that we need. So what we know about proteins, some of them perform almost all cellular functions, as you saw previously, even muscle motion, and some are responding to pressure, like heat or cold shock, sometimes heart events, and so on. Proteins are long chains made of amino acids. There are 20 types of amino acids. the fold of each protein, which is carefully designed for fulfilling its function, is determined by the sequence of the amino acids that are building it. So what I mean, what I want to tell, is in nature, structure means function. Look at this machine, very sophisticated machine that is called paperclip. It works only when the fold is correct, where the structure is correct. If it's being modified by fingers, without heating, without cooling, without cutting, without anything, just changing this structure or the fold, there is no function. And this is the way proteins work. So as I said earlier, there are 20 types of amino acids, and their sequence within the proteins, which enable its functionality, is dictated by the gene that codes for proteins. So proteins are made according to their genes, which are in the DNA. Actually, the DNA is a string of instructions written in a 4-letter language in which each 3 letters, or we call them also bases, are coding for a specific amino acid. These are called codons, these 3 letters, these triplets. So this is a typical enzyme. It's made of many atoms, many amino acids, each made of atoms. It has a active site. And if we look into the active site, this is here, and this is supported by the structure around it. I rotated it and I enlarged it, and you can see the active site now with the substrate. And you see that the substrate fills it exactly. If it was too large, it won't fit. If it was too small, it will flip around. So it must fit this exact. So this must be exactly according to what nature does. In this case, what nature needs is this for this substrate. So how do the proteins, are they made? Here there are 5 amino acids, 5 components. All of them have, it's part of the 20, yeah? All of them have the same backbone, and all of them have the backbone that can fit each other, and each of them has a different side chain that gives them the biological or the chemical character, and some of the side chains can also be biological tools. They get together, they become more and more and more together, so long that I didn't even draw the details. Then they become long protein. About each normal proteins are about between 200 to 500 amino acids, and it's worth nothing unless it is folded with an active site, as you could see earlier in the real protein. So this was known when I started my work, but it was not known how it happens. This is what I wanted to find out, how this happens. So many other people wanted to do it, and they were more experienced and they had better labs, and they failed. So there was a big skepticism about the possibility to understand it. There were even papers saying it cannot be done. Yet I tried to do this. I will tell you afterwards why. I knew that we also can fail, but I knew that if we don't fail, if we make it, we will have a big smiley. And this is what we have today. So the central dogma says the DNA that you can see here is where the bases are. It means the letters, the triplets that code for the proteins. The information is inside here. These are the bases, these are the letters within the double helical structure. And here it is not available for reading. It's covered, it's hidden. So the first thing that happens, there is a transcription, to a very similar molecule that can exist as a single chain. So here the letters are exposed and can be used. They are available for using. And this can be translated to growing proteins by a particle within every cell that is called ribosomes. Actually, there are many ribosomes within every living cell. So they are universal. It means they function in a very similar way in all cells, regardless of the source. It can be bacteria, it can be flower, it can be cockroach, it can be human. They do the same, the decoding and the production of the protein the same way. There is a huge number of ribosomes that function in every living cell. Even in bacteria, they can reach 100,000 when they work. And in some mammalian cells can contain millions. For instance, the liver may have 5 to 6 million ribosomes. In vivo, the ribosomes act continuously. They can form up to 40 peptide bonds in a second. They hardly make mistake. So I, as a very good and fast student, needed 6 hours for one bond, but they can do 40 in a second with about mistake rate of about 1 to 1 million. So now that we know how they work, we like to describe them as a factory that we call ribosomes that is made of 2 floors. In the top floor, the information is being decoded. The triplets are coming in. It means the whole messenger RNA comes in, and triplets, when a triplets come here, it's found what it is coding for. The amino acid that is cognate to it will come inside into the factory and be connected to the growing protein that goes out. This arrow had to be inside, but I don't know how to draw it, so I drew it outside, but it happens in the factory. Then this track will become empty and join the other empty tracks. And as I told you, there are 2 floors. Here the decoding is being made, and here the making of the protein, the making of the bond, which is called peptide bond. So actually what you saw in the factory is shown here now in molecular way. Ribosomes, all ribosomes are made of 2 subunits, small and large. These 2 subunits are independent in the cell. They live as small and large. They come together when ribosome has to work. And they are connected by contacts between them and by the track, the tRNA molecule that brought the amino acids. So here it is, and it connects between the decoding center and the peptide bond formation center, which is called PTC. So this is what we found after 20 years minus 2 weeks of work, and we made a little movie that is available in YouTube that you can see here. Here, messenger RNA comes to the small subunit that waits. There are factors in the cell that are coding that are telling the ribosomes that has to start to work. There are initiation factors. In bacteria, there are 3 of them, and what you see here is actually ribosome from bacteria. And the whole thing is monitored by them. So yes, there are many more factors. You will see some in a minute. So messenger is reaching the small subunit that takes it. We made the movie, us, together with 2 students of the Art Academy in Jerusalem. And the messenger now will go into its path in the small subunits, the top floor of the factory. And it's monitored by initiation factor 3. When this is okay, initiation factor 2 is bringing it the first track, the tRNA molecule with the amino acid, this one. And now where everything is okay, the factors go out and the large subunit can come and associate with the small one by their surface and by making bridges. Now the ribosome is ready to work. We look at it from the solvent side. Up is the large subunit, below is the small one. And you can see elongation factors bringing it inside the tracks. The tracks are being decoded, and peptide bond is being made. And you can see the ribosome helps. And it makes very fast, 40 of them in a second. So you can see here how the decoding happens in the small subunit. And from the large subunit, we left only the tunnel through which the protein goes out. So this is how it is being protected when it's made. Now you can see the whole ribosome again. The tunnel is inside there in the large subunit. And the process continues until the protein is large enough. Here it is being grown and coming out. And when there is a stop codon, a codon that says stop, then instead of tRNA comes a factor, recycling factor and release factor. These factors separate between the small and the large subunit. The protein can fold and come out and has to do what it has. And the tRNAs, the tracks, and the messenger and the ribosome are waiting for the next job. That's all. That's the whole story. So what we saw in the movie was rather fuzzy, but what actually happens is that we know the position of each and every atom in the ribosome, small subunit, which sediments as 30S and large subunit is 50S. You can see that the ribosome is made of many components. The main one is ribosomal RNA, another RNA molecule, and many ribosomal proteins in many colors here. And just to remind you, the small subunit is doing the decoding, and the large is peptide bond, is for peptide bond formation. Now let's look at these people. All of them are giants, either in music, like Mozart, Chopin, Schubert, or very intellectual writers. And what's common to all of them is they died very young. You can see their lifespan. The oldest is 47. Also, what's common to all of them, they died before or at the middle of last century. What you cannot see in the picture, but I can tell you, is they all died from infectious diseases that people don't die now from them. So what happened in the middle of last century? In the middle of last century, antibiotics came into use. And they saved the lives of many, many people, and of course, extending lifespan. So because of the fundamental role played by ribosomes, many antibiotics target them. Almost half, over 40% of the clinically useful antibiotics target protein biosynthesis, mostly by paralyzing the ribosome. What are the natural antibiotics? They are the weapons that bacteria from one type use to interfere with the life of other species of microorganisms when they have fights for space or for food or and so on. Question is, how do the tiny antibiotic paralyze the giant ribosome? So bacterial ribosome have a molecular weight of 2.5 million Dalton, and antibiotics less than 1,000. And the trick is that they are bound to the active site. So you see here the small subunit and the large subunit, and the circles are showing where are families of antibiotics. It means the natural antibiotics that were modified by companies. And you see all of them are bound to functional sites, either to the decoding, or to the bonding, or to the tunneling, or to the hinging. These are the motions that are needed for continuing. So this is the trick, and we want to show you a movie that was made based on the previous movie, and it's already 13 years ago, and as I told you, in YouTube, and it shows that the tricks played by the antibiotics. So this is a small one, edain, that doesn't let the messenger bind. That's all. Second one that you will see is tetracycline. It also works at the small subunit. It doesn't let the second tRNA bind to the second position. So one is bound, but the second cannot. Second position is called the A site, for those that want to know. So just occupies it. Then I want to show two that work on the large subunit. First is erythromycin, which is one of the first that came into use in medicine right after penicillin. It sits in the tunnel. If you remember, the tunnel that protects the protein and doesn't let more than a very short protein to be made, and then it blocks. And the last one that you will see is disturbing the formation of the bond, very clever chemically, maybe the most clever chemically. So here the bond is being made, and it just sits in the bond. No bond, no more protein. That's the whole story. So, sorry. All ribosomal antibiotics bind to ribosomal functional sites. We just saw it. But these are highly conserved because all ribosomes work the same way. I already said it. And mandatory for clinical use is the distinction between the patient and the pathogen. So how does this happen? This happens by subtle differences. Even in highly conserved regions, there are subtle differences. And I want to show you one to explain. So if we look into it, we look into the whole large subunit. This is the entrance to the tunnel, and erythromycin blocks most of it, enough not to let proteins go through it. So if we look at this position in the tunnel, like a perpendicular to its long axis, we see that there is a pocket, and the pocket is made just from ribosomal RNA. And all the members of the erythromycin family, which is called macrolides, are bound here. So some are bound excellently, some are bound very good, some are bound just good, but all of them block the tunnel. And all of them interact with one of the components of the tunnel that I marked as pink, and it's adenine A in position 2058, according to E. coli. So this is correct for all bacteria. All eubacteria have A in this position. Let me show you how it binds erythromycin. So this is this A, and this is erythromycin, and the affinity here, the chemical affinity, is very, very good. It means this is very cleverly designed by bacteria. Now, please, ladies and gentlemen, pay attention to this point, and you will see the difference between bacteria and us. This is all bacteria and us. But when there is a G here, us, there is repulsion. This is too close here, the distance, and there is no binding. That's all between bacteria and us, between bacteria and any higher organism. Second question that I want to discuss, second point, is resistance. Resistance to antibiotics is one of the most severe problems in modern medicine. And let's talk about it. First of all, how it is being acquired. So I will show you one of the methods. Selectivity was shown here between this and this. And bacteria can just change themselves to G. And now they are resistant. Actually, resistance to antibiotics is a basic process for the survival of many microorganisms, regardless of their exposure to modern clinical treatment. And this is a very important finding. And this is one of the most severe problems, as I said before. The World Health Organization said we are reaching post-antibiotic era. It means the time that antibiotics will not be useful anymore. And even the World Bank estimated that up to 3.8, almost 4% of the global economy will be lost by 2050 because of resistance to antibiotics. Can we combat antibiotics fully? In my opinion, not. Because bacteria want to live. Maybe want is a street word for it, but this is what happens. And they are cleverer than us in terms of survival. But we can do some things. So our lab decided in our lab to help it, because companies are hardly interested now in antibiotics. Most of the big companies are not interested at all. So until recently, all the known structures were of models for pathogens, not real pathogens. And this explained the common modes of antibiotic action, as I showed previously, but they do not explain species specificity of pathogens. And this is what we wanted to see. What's the difference between non-pathogenic and pathogenic? So we crystallized the large ribosomal subunit from a pathogen, actually a multi-resistant pathogen called Staph aureus. You can see here the crystals, and we got the structure. I won't show the whole structure. I will just show some lessons we studied by comparing it to non-pathogenic. So first of all, it's possible to increase antibiotic activity. More potent antibiotic can be made. We collaborated with a company called Nabriva, a small company in Vienna, and they made 16-fold stronger antibiotic. Lesson number 2 and 3 I skip. Lesson number 4 is beyond my expectation. Novel antibiotic binding sites. I didn't expect it. It's a very, very nice surprise, and in my opinion, also very powerful. So if we look here in the structure of the large subunit in gray, this is the skeleton in two views. This little bluish, this little cyan, show where there are additions on the skeleton of the large subunit. You can see some on this side and some on that side. And maybe you can see even better here from all sides. We drew here only those that are on the surface, and those that exist only in Staph aureus, not in the pathogen models. So we suggest to use them as new positions for potential binding site, antibiotics potential binding site. And I want to show an example here. This is part of the ribosome. There are here two structures, one of the model and one of the pathogen. Here they are indistinguishable, but here there is a little addition. This is what you saw in this view. And we suggest that this can be potential binding sites, because it's only in the pathogenic, and it is, we found that it is also important for protein biosynthesis. So actually in a model system, we identified 25 new potential binding sites, unique, and 16 of them where they were blocked chemically, they inhibit protein biosynthesis. And because these sites are not involved in the primary ribosome activity, namely in the decoding and peptide bond formation, no pathogen contains genes for their modification. It means the bacteria did not understand now, although they are clever, did not understand now that they are important. So for now, they can be exploited for the design of specific advanced degradable antibiotics, hence environmental friendly. So I didn't talk about, I talked a little about specificity, I'll talk about it a little bit more, and about degradability. It means environmental concerns. So first, specificity. I hope that you all know about the microbiome. The microbiome is where trillions of bacteria reside in our body. They are called the good bacteria. They are usually in semi-exposed organs like guts, ears, lung, and skin, and they are mainly harmless. But they are susceptible to antibiotics. And so if antibiotics can penetrate into where they are, they can kill them too, and then people can catch other diseases. Therefore, we suggest to exploit these sites and design those that will not disturb the microbiome, the good. Also, they can be degradable. What do I mean in degradable? This is the environmental and ecology consideration. This is based on the fact that most of the ribosomal antibiotics that are used today are extensions of small organic inside the cores that are not digestible, and when they go out from the body with the rest, with the sewage, they are not degradable. And therefore, they can penetrate into the water. They are too small to be used, to be found by most of the used facilities for purification. And then they go into the water. They can penetrate into the irrigation water, and they come back to us. So this is why when we design our antibiotics, we want to make them degradable, biodegradable. So now I want a few words, how did we start? Because I thought that it has some lessons that we can learn from it. First of all, why did we use X-ray crystallography? Because human can see, I think, maybe Insects. With normal microscope cells, bacteria, viruses, but not atomic detail. For this, we need crystallography. For crystallography, we need crystals. They diffract. We see the diffraction here. They diffract according to the Fourier transform, and if we collect the whole three-dimensional, whole space around the crystal, we can get enough information to build back the structure. So here you can see X-ray coming. There is here a crystal. We collect the data with computers and with knowing how to do it. This little thing is the crystallographer. It means like something like me. And then we get the structure. So we can get structures of many types of molecules. But we need crystals. Let's define what crystals are. So this is sodium chloride. It's only 2 atoms, sodium and chloride. And it likes to make a three-dimensional periodic system, even in nature, spontaneously. So you can see full periodicity in 3 dimensions of chlorine and sodium in between. This is a natural crystal. This is a crystal of a protein that was first crystallized in Oxford in the 60s, but a 6 grader, about 12 years old girl, could repeat it. So proteins can be crystallized by this very complicated, now we know very complicated structure that you know. But even before the structure was known, there was a big question mark. Actually, this question mark said no. And the people that tried to crystallize it, they explain it by all types of high degree of internal mobility, flexibility, structural heterogeneity, chemical complexity, all correct. But they paid attention less to a very important property of ribosomes, that they have marked tendency to deteriorate. Even when we prepared it, they deteriorate, and we get less than half of the expected activity. And I was much more open to this problem. So I thought, first of all, we have to find ways that the ribosomes won't be deteriorated when we made them. Okay, I thought about it, but I didn't know what to do until I read a paper about how is the metabolism of the sleeping bears in the North Pole. The North Pole, the bears are going to sleep for the winter, and a delegation went to see what happened inside them. So they found out that in every cell, inside the membrane of each cell, the ribosomes are fully organized periodically. Monolayers, not real crystals, but monolayers of organization, and this shows that ribosomes can be orderly packed. Every bear, every cell. So then I thought, why is it? Why did nature provide this mechanism? And in my opinion, this is the way to maintain a pool of active ribosomes for the end of the winter, when the bears would like to do other things and they need proteins to do them. So my conclusion was that stress induces periodic packing of ribosomal particles. So I thought, let's look at stress, at bacteria that grow under stressful conditions. I didn't want to work on the bears, on the ribosomes of the bears. First, I didn't want to kill more bears, and more important, because the ribosomes from bacteria can be useful for medicine, whereas those of bears not. So the solution was to use ribosomes from bacteria that grow under stress, like in the Dead Sea, in hot springs, or in waste of nuclear facilities. I just want to show you the Dead Sea. It's the lowest point in the world. It's in Israel. You see, from top, no animal, no tree, no nothing, no life. You can see from closer, these are all structures of salt that came out by evaporation. No animal, no plant. But there are 2 types of bacteria, and I want to show you the colonies of one of them, Heloarcula maris morti. Here, you can see it here too. And I knew about this bacteria, and we used its ribosomes. Also, we use ribosomes of Deinococcus radiodurans, which is a bacteria that can grow under super extreme conditions, very hot, very cold, environment, dust, no food, even under irradiation. And we got beautiful crystals. Look, how nice. So we got actually 25 types of them. I show here only 6, those that were useful for crystallography. And I want to show you 6 years after we started, we went to measure them. We took crystals to measure in this accelerator, which is in Grenoble. In Israel is a member of it. Actually, we used another one, but it's such a beautiful picture, I decided to use this. From top, it looks like that. Look how wonderful. From getting close, you see how it works. So here there are particles that be an accelerator, then they run around in this ring. And in the tangential, we have stations. Here there is a station that Frank is measuring on ribosomes. So each station has its own properties. Some are useful for ribosomes. So in 1986, 6 years after we started, we got the first diffraction. Look how beautiful, beautiful spots all the way to the end from the last subunit of halo arcula in time equals 0. We were really very happy, but not for long. Second picture was already deteriorated, damaged. It means the X-rays damaged the crystals. We repeated it 100 times, and we didn't get anything better. And we felt like we started from here, and we saw only this mountain. We call it the Everest, sorry. And we climbed it, and climbed it, and climbed it. Took 6 years until we got to the summit, and what did we see then? Not a really big, much bigger Everest, much taller Everest behind, that we will have to get there in order to measure. Much more difficult than until then. So this was the time I thought maybe we should stop this project, but instead I thought about why does it happen? All living tissues suffer from X-rays, but this is much too much. So I explained it by the crystals being weak or very soft, and the damage occurs in 2 steps. The primary is local disruption of the bond, and then when the bonds are being cut by the X-ray, the 2 parts of the bond are unhappy, going around and break more bonds in order to satisfy themselves, either to give an electron or to take an electron. And we thought that maybe the way to stop it was to cool the crystals so cold that there will not be energy for the running around. And this worked, although everybody didn't think so. Even I. Look, this is the experiment done at Stanford. Look at my face. Even I didn't believe in our own experiment, but it worked beautifully. Have a look. And 20 years later, look how happy I am. Now it works. And this is the machine that cools, and this is the machine that measures the X-rays. This is in Grenoble. Actually, not only for us, it worked for the whole world. We introduced it here at the end of 86. Within the beginning of 87, 90% of the world used it, and now everybody. So what you see here is the number of structures that are known now and have been deposited in the protein data bank, and this is the year. And have a look, this is only till 2011. 47,000 structures. All of them done, or 99% of them done in our cryo method. And all of them are of very important materials for medicine or for making new materials. Actually, now it's 2017, and there are 80,000 new structures that couldn't be done otherwise. So how about future? We go back to the lecture, and what I want to tell you, we plan to identify the specific structural features which are unique for each pathogenic bacteria. What I showed you about Staph aureus, we want to do for all. Once we identify such features, we will design novel antibiotics for them. In these studies, in the novel antibiotics, we shall use cryo-electron microscopy, which provides high-resolution structures with no need of crystals. All what I told you until now about crystals, we will not need them in the future. Taking advantage of the current revolution in biostructure research. This is our program, and I hope that you support us. So before I finish, I want to thank the Weizmann Institute that let me work. Here is the president that made the decision, Michael Sala. He was a biologist. Although I was called the dreamer of the world, he still supported me. And when he finished his term, he was replaced by Chaim Harari, who is a physicist, and he also supported me. They were encouraged by the Scientific Advisory Committee, among whom I want to highlight Alex Rich, who tried to crystallize ribosomes with no success for 10 years, and he was still very supportive. Actually, the whole work started with a very strong collaboration with Max Planck in Berlin, with Dr. Weizmann, that he really wanted to see the structure of the ribosomes, and he helped in establishing a group for us in Hamburg, near the synchrotron there. He died 10 years before the structures came, unfortunately, and then he was replaced by Franceschi and then by Fucini. So I want to thank my research group in the two places for their devotion and determination in good and in bad times for many years. Here, this is the group in Hamburg. It was terminated in 2004. It always had angels. The technical assistants say that otherwise they cannot work if they're not angels. Ribosomes are too complicated. And the group came to the Dead Sea to look for new bacteria. Well, they had good time. The group in Israel is still working. Here you can see people that came and went through the last 3 years. Some of them are still with us. I want to highlight Dr. Anat Bashan, who is the senior scientist that runs the show all the time when I'm doing other things. And I cannot show you one by one, but I want that you pay attention to Tamar. Tamar came 16 years ago for 10 weeks. She's still around. At the day that I took a picture, she had birthday, so she baked a cake. You want to see the cake? Here is the cake. And it shows that in my group, ribosomes are considered sweet. I also want to thank my family. I cannot show you all of them. My mother, my sister, and so on. Just my daughter, who is a doctor, a medical doctor, and her daughter. This is the girl that crystallized the ribosome when she was 12. And they were also supportive all the time, even when I wasn't there. My granddaughter actually gave me a speech and then a prize. I will show you the prize in a minute. In the speech she said, as you know, she is a very busy scientist, but she always finds time for me. And this is the prize that she gave me. No, not this one. This is the medal, Nobel medal. Her prize is. The grammar of the year is Ada Yonath, at least as important. And when I asked her, what is the year? Every prize has a year. She said, you have to reprove yourself every year. I'll take it off the wall if you fail. Still on the wall, you see? So ribosomes became popular. In a carnival, he is me. And she, his wife, is a ribosome. Look, protein comes out of her. Also, younger children became ribosomes. And this is what happened to me by one of the best artists and writers in Israel, Michel Kishka. And if you have a look very carefully, this is the large subunit, and this is the small one, and it's symmetrical. And he thinks that the best place for antibiotics is here and here. Thank you. Okay, I hope that you liked it, and I hope that something will come out of it, at least mentally, intellectually. And as I said, I would have liked to be here with you today, but since I cannot, good day to all of you.
Interesante video de la doctora Ada Yonath. Es también más interesante que cuando la contactamos. Debo decir que me sorprendió, pero ella recibió la medalla Vascoño de Balboa, pues un máximo honor que puede quedar la presidencia pues, a ciudadanos extranjeros. y a panameños en Panamá. Así que nuestra invitación fue realmente calurosa, digamos, y ella dijo, Panamá, claro, sí, yo conozco Panamá, he ido a Panamá, y sobre todo, pues, tenemos que decir que pues, la charla es súper interesante, y al final el mensaje que nos dejó es básicamente seguir intentándolo, seguir buscando. La ciencia es bonita y la ciencia da resultados, obviamente 20 años después, como dijo ella, ¿no? O mucho tiempo después, mucho esfuerzo, mucha gente, muchos colaboradores. Ahora quiero pasar a leer la información de la doctora Shulamit Levenberg, que es otra invitada, otra de nuestras invitadas de parte pues, de la Embajada de Israel y también pues, invitada pues, del Congreso. La profesora Shulamit Levenberg es profesora en la Facultad de Ingeniería Biomédica del Technion, Instituto de Tecnología de Israel, y está trabajando en básicamente lo que es células madres y ingeniería de tejidos. Bueno, entre otras cosas interesante, y vamos a verlo en la charla también, es que también esta parte de Biomedical la ha llevado también a la industria, y algo de esto que hablaba pues el embajador Red ayer sobre cómo es la mezcla pues de universidad y empresa. Esto es un gran ejemplo que vamos a ver con la doctora Shulamit. Bueno.
Hello, my name is Shulamit Levenberg. I'm from the Biomedical Engineering Department at the Technion, and I would like to describe to you today some of our studies on tissue engineering and regenerative medicine. I will focus on our approaches for bioprinting vascularized tissue construct. So we know that there is a shortage in tissues when a tissue or an organ is injured or is not functioning. So how do you repair the tissue? How do you repair the function of the organ? So one option is to take from donors, and there is a shortage in donation. And also the patient will have to receive immunosuppression through all his life. Another option is to take from the patient's own cells, own tissue, to take from one area to repair another area, but this will cause two sites of injury and complicated surgeries and a lot of pain for the patient. So tissue engineering evolved as a way to try and solve this problem by isolating cells from the patient and then culturing them in vitro, expand the cells because you need many cells to engineer a tissue and organ. And then one can seed them on 3D scaffold. Usually it can be from biodegradable material. You can imagine a sponge or a gel with the cell attached to the sponge, they grow in it, and then they can differentiate to form the tissue that later can be implanted to repair the damaged tissue. So since we know that in each tissue you have more than one cell type, also in the lab when you engineer a tissue, you can take a mixture of cells. For example, if you want to engineer muscle tissue, one can take the myoblast, the endothelial cells, they form the blood vessels, support cells, and mix them together. Of course, you need to find the right cell ratios and the right medium composition, but then one can take the mixture of the cells and seed them onto the scaffold. And here you can see a sponge-like scaffold where the cells will be seeded into the pores of the scaffold and will start to organize themselves into the tissue. And we know that after implantation, the tissue needs to get oxygen and nutrient very fast. So we found that if we add endothelial cells to the culture, they will assemble into blood vessels in vitro, and these vessels can help later after implantation. So the cell will form the tubes. You can see tubes that are connecting. And if we will add support cells, some of them will differentiate to smooth muscle-like cells or pericyte cells and will be recruited to the vessel wall and will stabilize the vessels. And if we add myoblasts, they can differentiate into myotubes and form the muscle fibers. And by this, we can get engineered muscle tissue, which is fully vascularized, and you have the blood vessel network in vitro. So this is how it looked under the microscope. You can see the endothelial cells here are fluorescently labeled in red. Everything else is filled with cells, you just don't see it. But here the endothelial cells are forming the tube, and one can really follow by time lapse. You can see how the cells are organizing themselves, how they're forming the tubes, and how in a zoom in, you can really follow the tip cells that are finding the way through the matrix and through the other cells to form the tube, and then also to connect to the other tubes. So in the lab, some of our studies are focusing on trying to understand the mechanism of this vessel network formation And here, for example, you can see studies where we study how flow can affect the vasculature. And specifically, you can see here a demonstration of a bioreactor that Barak Zohar, a PhD student in my lab, developed. And in this very unique bioreactor, one can apply flow on the cells and really visualize it at the same time under the microscope. And then one can track the vessels, the dynamic, and you can also engineer one large tube and analyze how the cells now will line the walls of the tube under flow. So these studies are really, they're interesting and give us some insight about the mechanism, how the vessels are being formed and what angle they take and in what speed they form the tubes. Another parameter that can affect the vessel formation in vitro is tensile forces. In our study, we wanted to see how it can affect the blood vessel formation. So we can take as a control the scaffold where the vasculature is forming in all directions. And then we can put the construct under static stretch, meaning one stretch, or under cyclic stretch where there is ongoing stretch all the time. And we wanted to analyze whether it will affect the direction of the tube or alignment of the tube. So what was interesting to see, and this is a study of Shira Landau in my lab, she found that the vessel orientation is really happening by a force-induced anisotropy of the underlying scaffold, meaning that the cells will see how the fibers of the matrix align because of the stretch, and the vessels and the other cells will also align in this direction. So the alignment will be in the direction of the force. And it was really very interesting to see the whole tubes now lining in one direction. And you can see here in another system, this is a study of Deckel from my lab, also a PhD student, and you can see the control where the vessels are going in all directions comparing to the stretch where the vessels are aligned in one direction. So in the cyclic stretch, what happened that the direction that the vessel took was actually perpendicular to the stretch direction. So, and this happened because of the tendency of the cells to minimize the elastic energy transferred to them by the scaffold. So here you can see that the vessels are aligned, so the tubes are really aligned, but perpendicular to the stretch direction. And what's happening if you start with static stretch and all the vessels are aligned in parallel, and then you switch to cyclic stretch? What we saw that if you wait enough, the tubes are really turning, and eventually they will go to the direction perpendicular to the stretch direction. So one can really use forces to direct the formation of the vasculature of the vessel network in the engineered tissue. And why it's important? For example, you can see here in the muscle, we have the vessels are aligned in the direction of the fibers. And what will happen if we now take the engineered muscle tissue and implant it? We can implant it either in the right direction or not in the right direction, or the tubes can be unorganized without any alignment. And what we're able to show if we implanted it in the abdominal wall muscle, and when we implanted it in the right direction, we could see longer engineered vessels which are now perfused, and you can see blood that is flowing through them, meaning that there is a meaning. It is important to align now the tubes and to implant it in the right direction, because then the vessels can anastomose and have a better connection. So if we have now engineered muscle tissue, it can be used to repair damaged muscle. And you can see here an example of repairing the abdominal wall muscle. You can take the engineered tissue, it can be vascularized with all the vessels that we engineer in the lab in green. And what was very interesting to see, that these engineered vessels really recruit the host vessel to penetrate into the implant. And not only that, they can also connect to the host vasculature. You can see here different type of connections. And the connections are actually very important, because then some of the engineered vessels become functional and blood start to flow through them, and blood will now penetrate faster to the inner part of the construct, and eventually we will get the whole implant fully vascularized by the host. And the engineered vessels really were very important because they facilitated the process of bringing blood and oxygen to the construct. And this is how it looked under the microscope. You can see the engineered construct, and you can see in green the vessels. And in blue, you can see the host vasculature now penetrating into the construct. And we can also inject here rhodamin dextran in red, and it will flow in the vessels. And every time we see the red, it means that the vessels are now functioning and blood is flowing through them. So in higher magnification, I hope you can really appreciate the connection, the nice connection that you can see between the host vessels in blue and the engineered vessel in green, really very nice anastomosis. And the next step was to see whether we see any of the red staining getting into the green area, into the engineered vessels. And indeed, you can see here, we have connections between the blue and the green vessels, and the red staining, mimicking the blood perfusion, is now going into the engineered vessel, meaning that they become functional. And this was really very exciting. So Shahar in my lab wanted to study whether it's important to mature the vasculature in vitro to get this perfusion and anastomosis. So she compared 1-day implant to 14-day implant and implanted them, and wanted to see whether we'll see more recruitment and more perfusion when the vessels are mature, as you can see here in the image. And indeed, this was very important, so she could compare the different time points in vitro and the different time points in vivo. And what was very interesting to see that indeed, culturing the cells in vitro, the time in vitro was very important to enhance the maturation of the vessels in vivo. And what was also very interesting to see, that the penetration of the host was also affected. So you can see here the different time points in vitro. It was grown in vitro only for 1 day, or to 14 days, where the vasculature is really mature. And then looking at the different time point in vivo, and looking at different region of the implant. So the R3 is really the deeper area, and it will take longer time to reach the inner part. And we wanted to see if we can make it faster by culturing the implant for longer time in vitro and maturing the vessel network. And indeed, in the 14-day core culture, you can see more host vessel penetrating into the region, into the deeper region, into the R3. And we could see also very nice connection, different type of anastomosis between the host and the engineered vessels. We could see here end to end, but we could also see mosaic connection and wrap, meaning that now one vessel is wrapping the other. And not only connection we could also see that now the blood is being perfused into the engineered vessel, and you can see here in red, and we can quantify, and we could see that we have more perfused vessels when the implant was cultured for longer time. And what was also interesting to see, that we saw more blood clot formation when the implant were implanted very early, only after 1 day culture. And this led us to the assumption that the blood clots that are formed when the vessels are immature, preventing the flow and the perfusion through the engineered vessels. And indeed, we could see that the implants that are immature, expressing high level of factor that are inducing clots. So we could now see that we can culture endothelial cells and support cells and form the vessel network. And we can also add the tissue-specific cells. For example, I showed you the skeletal muscle tissue that is formed. And we can also use it in other models. For example, here we took cardiac cells and co-culture them with the endothelial cells, and we were able to get vascularization of the cardiac patch without affecting its beating, spontaneous beating. And also in the pancreatic islets model, we could see here islets that are secreting insulin in response to glucose. And now we can seed them and embed them within this vessel network, that we were able to show that really increase the survival and the function of the pancreatic islets. So in these three models, we could also show that the vascularization in the lab helped the vascularization in vivo, help the integration and the perfusion that we can get in vivo. In the abdominal wall muscle that I showed you before, you can see it here, you can see the graft area and the blood now penetrating into the area. And we could see it also in the two other models, in the cardiac patch that we implanted onto the heart. When it was pre-vascularized, it integrated better. And also in the pancreatic islets, when we took relatively small amount of islets and implanted it under the skin, if the cells were pre-vascularized, then they can rescue the diabetic condition in the mice. But in all of these cases, it's a graft, and we have to wait till the host will send the vasculature into the implant area. So we wanted to move to more complicated cases where you cannot rely on the surrounding to send the vasculature inside. So we wanted to engineer tissue flaps, meaning that the tissue is coming with its own vasculature, with its own blood supply, and you don't have to rely on the surrounding. So what we did here, we took the engineered construct, pre-vascularized, wrapped them around AV bundle, and the hypothesis was that we will get sprout from the AV bundle, feeding the implant. Later on, now we can take the implant and transfer it as a flap to a full thickness defect where we cannot rely on the surrounding, and it will survive because it's coming with its own blood supply. So here you can see that indeed we could see sprouts coming from the AV bundle to feed the construct. And this is a very nice image done by Idan that really mastered the micro CT to allow us to show all the details of the vasculature that is now sprouting from the AV bundle to feed the implant. So now when everything is fed by the artery and vein, we can now take the artery and vein, cut them and flip the construct to another area. And you can see it here. And because the construct is coming with its own artery and vein, already after 1 day, it's fully vascularized and you have blood flow very nicely into the construct, meaning that we were able to engineer tissue flap in the lab and using one step in vivo before a transfer of the flap. So we wanted to take it another step and see if we can combine now hard tissue. So we have composite tissue of soft tissue and bone tissue. So we have the soft tissue here, as I showed before, and now we can add the decellularized bone matrix, and we wanted to see if we can get sprout coming from the AV bundle to feed the bone. But later, we wanted to see if we can use it to repair a bone defect. So you can see it here. This was done in collaboration with Gordana Vunjak-Novakovic from Columbia University, and she sent us the bone, the decellularized bone construct. And you can see here the AV bundle and the soft tissue and the hard tissue. And you can see here how we can see sprout from the AV bundle reaching the area of the bone construct. And here you can see it really very nicely. I really like these images. You can see the bone and you can see how the sprout from the AV bundle are going all the way and penetrating the construct, especially when it was pre-vascularized and provided signals from the sprout, from the AV bundle, for the AV bundle to sprout into the bone construct and feed the bone construct. So these are really amazing images. And we wanted to see now, okay, we have the decellularized bone matrix, whether we can now add osteoblasts and really form bone tissue that we can use to repair the bone defect. So we took mesenchymal stem cells and differentiated them to form bone, and you can see all the markers here. And indeed, when we added cells, the osteoblasts into the construct, they provided more signals and we could see more vessels comparing to matrix without cells, meaning that having the cells there provide more signals for the vascularization of the construct. So we have the construct and we see that we can induce sprouting from the AV bundle to the area of the construct that make connection with the self-assembled vessels that we can form in the tissue. And we wanted to ask if we can now mimic everything in vitro. So we won't need this step of AV bundle, wrapping the AV bundle and sacrificing the AV bundle. We wanted to see if we can do everything in vitro, engineer everything in vitro, including the large vessels. So the concept is that we thought was we can engineer the larger tubes, we can also print the larger tubes, and the tissue around the large tube can be seeded with endothelial cells and support cells that will form this network. And the hypothesis was that similar to what we saw in vivo, we could enhance, we can provide signaling to the large tube to enhance sprouting from the large tube to connect to the small capillaries. And if we flow medium through the large vessels, they can reach the small capillaries. So this is what we wanted to engineer. Engineer the large tube. When we have flow inlet, we can perfuse, and we have the flow outlet, and then we have the endothelial cells and support cells that will form spontaneously the vascular network, as you saw in the animation. They will provide signaling to the large tube to send sprout and connect, and then if we flow through the large tube, the medium can reach the inner part of the construct, and we can get fully vascularized tissue. So at the beginning, we thought that it's better to give some cues to the cells. So here is the large vessels, and we thought that we should make some openings to induce sprouting from the large tube to the surrounding, where we will have the self-assembled capillaries by co-culturing endothelial cells and support cells. So Barak decided to take this challenge, and he decided to design the angiotube, when you have large tube and you can induce the sprout by providing openings, these holes, for the cells to sprout out. So this is the design and the fabrication. He used the PLLA macrovessels with laser drilled microchannels, and the size of them, you can see the range from 50 micron, the inner part, and going to the 170 micron. And this is based on other studies showing that this will help the endothelial cells to form the tube. So you can see here some of the design, the characterization, the flow, velocity distribution, and the pressure and the stress that we have engineered. So they really like this modeling, and this model show us all the flow parameters of the tube and what the cells will experience when they're in the tube. And here you can see the seeding of the endothelial cells, and you can start to see that indeed through the opening, the endothelial cells will sprout and start to form the tube that will go out from the macrovessel to connect to microvessels. And you can see here the self-assembled vessels in the tissue around the tube, and we use 3 different models of the tube related to the tissue around it. And the endothelial cells in the tissue around the tube are labeled in red, and the endothelial cells in the tube are labeled in green. So we could see the sprout that are coming out in green and connected to the red vessels that are outside. So now if we flow medium through the large tube, it can reach the smaller capillaries that are in the tissue around the tube. And this was really very nice to see that if we inject now a blue dextran, we have, and we check the patency of the tube, so it really don't leak. And you can see the blue is going out only through the vasculature, through the small sprout that are going through the holes of the tube, and eventually they will connect with the red one, so we'll get blue staining also in the red small capillaries. And then Barak and Lior wanted to take it another step and see if they can really engineer a tissue that can be implanted. So they design a PDMS mold that can later be broken, and you are left with a tissue that you can now implant. And in this tissue, they use 2 rods. They engineer the tissue here, and after the tissue is being assembled, you can take out the rod and line the endothelial cells in the larger tube and try to see connection between the larger tube and the sprout that will go outside. And you can see here that indeed you can see sprouts coming from the larger tube and reaching these small capillaries, similar to what we saw before. And we can see very nice anastomosis, very nice continuation and connectivity between the green and the red, and different type of connections, similar to what Shachar saw in vivo, the end-to-end connection, the mosaic vessels, and the wrapping. But now we see it in vitro. Now we engineer this multi-scale vasculature in vitro. And this was really very exciting for us to see. And if we inject the blue dextran, the vessel is intact, not leaky, and the blue dextran will go only through the sprout and will go after connection between the sprout to the self-assembled capillaries. We can see the blue going also into the smaller capillaries and feeding the implant. And now we can break the PDMS mold, and we're left with the tissue. And we can compare tissue with the multiscale vasculature and the tissue only with the small capillaries. And we were able to show that indeed, where you have the multiscale vasculature, it's really helping to vascularize the tissue, and we get more penetration of the host vessels into the implant, and we see more perfused engineered vessels when we have the multiscale vasculature, as you can see here, comparing to just the smaller capillaries. And more penetration of the host vessels into the area. You can see in blue the penetration of the host vessels feeding now the implant. So now we can make it more complex. We don't need only to have the rods. We can really print the tube and print the larger tube and the intermediate tubes, and also get the self-assembled vessel by now printing the tissue. So this is not a regular printer, and the bio-ink is not a regular bio-ink. The bio-ink is made from cells and biomaterial that can actually help us to engineer the tissue, the scaffold, and the cells, and we can use different heads with different cells, and we can pattern the tissue. And what you see here is an example of having the large tube, the large vessel, and we can see the cells in the lining the vessels and the cells in the tissue around it, and form the more complex tissue that will also mimic better the complexity of the vasculature. And we have here another printer in the lab, you can see here, and this is also using a different method of printing into the fresh bath, and you can see more structure of the vasculature that we can print, and these will be connected now to the small capillaries, so we can really feed larger tissue that can be used for implantation. So if we can engineer, if we can print the scaffold, we can actually decide what will be the geometry of the pores. So Ariel in the lab wanted to study whether the geometry will affect the sprouting of the vessels. So this is a collaboration with York Lahan from Michigan University. They send us scaffold with different pores, shape, geometries, the hexagon, the square, the circle. And Ariel developed the seeding techniques that allowed him to see vessels that are now lining the shape of the pore. And this is a starting point, so we get a starting point of a circle, of a square, and we wanted to see how it will affect the sprouting toward the inner part of the pore. And indeed, you can see that in different geometry, we could see different formation of the vasculature, meaning here we saw faster tubes that are now reaching the corners of the shape, comparing to circle when they kind of more circle around and less are found in the middle part of the construct. And what was interesting to see that the vessels were more prone to get into the corners of the shape. You can see here of the pore. And when we track them, indeed, they started from the side of the wall and reach the corners in the hexagon where we have more corners. So we had more options, more angles for the vasculature. And this could be explained by the fact that we saw more support cells concentrating in the corners. So you can see here in the corner, so you can see the peaks of the support cells and showing that you have the corners here and you have the 4 corners in the square. And this could explain why we have the tendency of the vessels to go toward the corner where they have more signals from the support cells that calling them to reach the corner area. So what we can do with the engineered tissue, for example, one area that we wanted to focus on was the spinal cord regeneration. And we wanted to see if we can engineer a construct that can regenerate the spinal cord. So you can see here an example of using a model of paralyzed rats. The rats cannot move their back legs. And we developed a construct with stem cells that can secrete neurotrophic factors. And when we implanted them into the paralyzed rat, we could see that the rats started to walk. And you can see they're standing on their legs and actually walking almost normal. And this was really, really amazing. We did not expect such recovery after implanting a tissue in the spinal cord, in the injury site. And this really enhanced axonal connection and axonal growth and allowed the rats to start to move the legs and walk almost normal. So Ben in the lab decided to design a construct by bioprinting, design construct that can help the axons to elongate. So he designed a very nice method of template printing where he could print the template, and he wanted to design it in very long channels that will help the axon to grow. So he used a template, and then he used the scaffold, and he added a step where he had a freeze dry when you can see smaller pores beside the larger channels, and this is the scaffold that he got. You can see the channel scaffold where you have the larger channels, and in between you can see the smaller pores that allow a medium to flow from one channel to the area, from one channel to the other, and also to allow cells to go from one area to another area. And because you do it by 3D printing, you can now look at the shape of the spinal cord, and this is an MRI image of the spinal cord of the rat, and then you can now print the exact shape of the spinal cord, the exact shape of the injury site, and this is what Ben did, and he implanted it into the spinal cord and could show nice elongation of the axons through the channels that he designed by the printing of the construct. Another two projects in the spinal cord area are, one is actually focusing on the vasculature, showing that we can vascularize the construct, and this will enhance the overall vascularization of the injury site. And this was very important, and we got a better sensory recovery when we induced this vascularization. And another important study is actually using, instead of using the cells, using the extra cellular vesicles that are secreted by the cell, the exosome. And when we modified the exosome to include the sRNA of P10, and by this we could induce regeneration, induce axonal growth, and we could get the rats to walk by giving the exosomes through the nose. It reached the spinal cord injury site and induced the regeneration of the spinal cord. So based on this finding, we founded the Neurexon, a company that is focusing on spinal cord regeneration using exosome modified with exoP10. And this is really, we really hope to be able to move it all the way to the clinic. Another important area is the skeletal muscle tissue, when we're trying now also to use it to fight disease, and in this case to fight diabetes. And in this, we now engineered muscle, and we also engineered cells to express high level of the glucose transporter. And we were able to show that this construct enhancing glucose uptake in the lab, and also after implantation. After implantation, we could see that if we implanted it in diabetic mice, the blood glucose levels will be reduced because we implanted the muscle with the cell that overexpressed the GLUT4 transporter. So this is really very encouraging, and we really hope to continue the studies and test them also in human cells and in human systems. The nice thing was that even if it was like just a small piece of muscle that was implanted, we could see a systemic effect of the GLUT4 overexpressing muscle, and we could see less lipids in the liver, showing us that the rats really are in better condition comparing to the diabetic mice. Also in another model where we implanted it, this is the model of insulin resistant model. So here also we could see that implanting the construct, the engineered muscle with the GLUT4 transporter, we could see that the construct stabilized the blood glucose level, follow a glucose stimulation test. So we could see that the engineered construct can be very relevant and can help in different conditions. We saw the spinal cord injury or the diabetic situation, and of course other systems were also shown. I mentioned the bone and the cardiac, but the engineered tissue can also be relevant to other areas. And one area that I want to demonstrate now that is different from the medical application, this is trying to engineer the tissue for food applications. So we did it in the lab. We took muscle cells from a cow and cultured them on edible construct, and we were able to engineer a piece of tissue that is really mimicking the real tissue, but now can be used as food, as alternative to the steak that is taken from the animal. And we were able to show very nice muscle formation using bovine cells, and using our technique of co-culture, adding endothelial cells and support cells, and the muscle cells on the scaffold, here edible scaffold, we could see nice formation of muscle that can be used also for food. And we're adding now to eat fat cells, so we can combine fat and muscle to get a real steak that is actually harvested without harming the animal. And based on this technology, we founded Aleph Farm, and Aleph Farm is now taking the technology to produce thin cut steak. This will be the first product of the company. You can see here some of it already cooked as a chef and plated and tested, and people say that it's really very similar to the original. And now with the 3D printing, we can also print different cells. We can print muscle cells and we can print fat cells. And this is a design showing a formation of a thick steak in the lab, which was also cooked. And this, we hope, will be the next generation of the product of the company. So based on the technology that was developed in the lab, we founded Norexon, Aleph Farm, and also NanoSynex, a company that is based on the microfluidic system that we developed in the lab, using it now for as a microfluidic device for microbial susceptibility testing, to be able to test much faster which antibiotics one needs when they get into the hospital with some problem that need antibiotics, but you don't know which one till you test it. And our device is now doing it in 4 hours instead of longer time in the current way. So I would like to end here and thank my lab, of course, the funding agencies and all our collaborators. And thank you for listening. Thank you very much.
Sí, gracias a pues, la doctora Shulamit, la doctora Yonath. Bien interesante la presentación, sobre todo porque nos habla de no solamente de cómo la ciencia básica y esos avances de la ciencia básica están pues, al alcance de todos, sino que los lleva también a un producto, la parte microfluídica y la parte entonces también pues, de tratamientos biomédicos. Quiero aprovechar este espacio para decir un poco lo que va a pasar en las próximas sesiones. Hoy tenemos un día bastante interesante y a muchos de ustedes les debió llegar información sobre esto, pero igual es bueno recordarlo. El día de hoy, aparte de esta sala magistral, tendremos conferencias en la sala de A, B, C, D, E, F, G y H en la tarde. O sea, en la mañana y en la tarde. Entonces, hoy me gustaría también decir que tenemos pues varios simposios, entre ellos el simposio de biodiversidad de hongos en la sala A, el simposio de investigación e innovación en programas de control biológico en Latinoamérica en la sala B. En la sala C tendremos la calidad de agua y posibles tecnologías de tratamiento en las áreas urbanas de Colón. En la sala D tendremos lo que es la mesa redonda 1, que es de educación a distancia en situaciones de emergencia. Y en la sala E tendremos lo que es bioética en la era del COVID. Y tenemos además 2 sesiones temáticas, 2 sesiones de simposio y mesa redonda, 2 sesiones temáticas. Una va a ser la biociencia de ciencias de la Tierra y ciencias de los materiales, que va a ser en el salón F y G respectivamente. Además de esto, lo invitamos a todas las secciones de la plataforma. Así son, entre ellas, pues la sección de pósters y la sección también de patrocinadores. Con todo esto ya está abierto, ya se puede interactuar. Una cosa más que quería decirle, pues recordarles pues, que ya se abrió lo que es el libro del Congreso. Ya se subió a la página web. Pueden ir a congreso.apanac.org.pa. Además de esto, yo sé que algunos han recibido ya el correo sobre los accesos a StreamYard. ¿Sí? Recordar que las charlas, si usted tiene una charla el día de hoy, hay que entrar por StreamYard. Se puso en la página web del Congreso lo que es un acceso, una guía del participante Sé que algunos ya la vieron, pero también se lo comparte a todos para que todos puedan mirar, primero que todo, un poco de las reglas del juego, un poco de cómo se va a llevar la dinámica y sobre todo los que están pues, en las sesiones como exponentes. Y del lado acá, la plataforma de Ferial, vamos entonces a estar acá haciendo preguntas y básicamente pues, usted puede navegar a través de todas las salas, buscar la que más le parezca y entrar en ella. De nuevo, ir a la página web, todo está en la página web, mirar la agenda de hoy, mirar el libro del Congreso que tiene todos los detalles. Además, estamos trabajando en una agenda un poquito más detallada que vamos a pasar el link pues, durante el día. Agradecemos entonces a todos los participantes. Sé que hubieron algunas preguntas y comentarios pues, en la sesión de chat. Estamos también tratando pues, vamos a responder esto cuando sea posible. Y agradecemos entonces todo esto. Tenemos una agenda bien interesante hoy. Lo esperamos entonces en las sesiones de temáticas, mesas redondas y simposios de hoy. Y en la tarde, pues entonces otra sala magistral, una sesión magistral y de nuevo entonces salas de mesas redondas, simposios y otras salas temáticas. Cualquier pregunta estamos a la orden. Así que nos despedimos esperando pues que a las 10, un par de minutos, comiencen pues las sesiones temáticas de mesas redondas y simposios. Gracias.